Welcome everyone, Dr. Shayne here. Hey, I wanted to switch gears a little bit on the topic because oftentimes I really get passionate about the microbiome and how that impacts our lives and what we can do about it. You know, the tools we use to modify and modulate the microbiome. One of the tools that makes a lot of this possible, and we've got different research groups here at the laboratory, which is where I'm at now. And one of the core sciences is what we call genomics in general. And genomics is the study of the genetic information that resides in all bacteria, all fungi, viruses, phage, humans, all animals, etc. So the power of genomics gives us so many insights. And in particular, nutrigenomics is the study of how nutrients affect our genetic expression, our genetic regulation at the cellular level. So literally at the level of the DNA, where it impacts the way we create metabolism, the way we modify our epigenetics, the way we regulate metabolomics and proteomics. These are all intimately related to the genomic profile and how it interacts with the environment. And in particular, the environment that we consider nutritional. So food and supplements is our particular interest. So I wanted to go over a bit on the concept of how we take this information, because we've spent a lot of time researching and understanding this nutrigenomic relationship between nutrients and our body and our cells and our genetic expression. One of the ways in which we look at this in detail is we look at something called single nucleotide polymorphism. So by taking a DNA sample of someone and analyzing for these polymorphisms, you look at what we call mutations in genes. And these genes in particular are genes related to nutrient responses. So that could be the methylation cycle, that could be vitamin D receptors, that could be fatty acid metabolism, it can be protein metabolism, amino acid regulation. These are all part of the health piece towards our genetic personalized journey. And the research we've done over the last, it's been over a decade now, using this equipment I have behind me right here. This is a really highly technical, what we call sequencer. So this instrument behind me, we take a sample, whether it be a swab or a stool sample or blood or any way in which we can get access to DNA. That instrument allows us to sequence either the person or the microbiome or an animal. I've had people request, looking at racehorses or dogs and cats. We spend most of our time, of course, in humans and microbiome studies. Once we have the sequence and once we can look at these SNP analyses, we can then start to extrapolate how this impacts our health. And the personal genetic journey each of us are on isn't simply about our macronutrients, our proteins, our carbs, and our fibers, etc. Oftentimes it's about the detail, the small micronutrients that can be the phytochemical compounds. It can be the metabolites that our microbiome makes based on what we eat or what we supplement. And each and every one of these genes in our body has a purpose and a response to nutrition. So if we look at an individual's or a group's nutrigenomics, we can then analyze and say, okay, this group or this person in particular has what we call weaknesses in their genetic profile. These are predispositions to not being able to bind as much B12 or folates or assimilate these things or enzymatically modify and convert these nutrients into what we call, let's say, methylation compounds, or the receptors on the cells that respond to vitamin D or vitamin A or vitamin K2. If these receptors are somehow genetically mutated or altered, they don't respond well to these vitamin D, K, A, or E responses. So we have to modify or change the supplementation or the diet to overcome what we consider essentially a genetic weakness in many ways. And that's why we do this research, so we can personalize the supplementary, and then a clinician can take that a step further and start to personalize dietary or even lifestyle changes that is designed specifically around someone's genetic background. And oftentimes that genetic background, of course, is passed on. So in that sense, it allows a clinician to not only modify that individual, but then have in their mind, when they do further studies in family members, they can continue to modify in a very personalized way what types of nutrients this person may need, what types of enzyme support, what types of membrane support, what types of DNA damage support, etc. These are things that we can learn from performing what we call nutrigenomic or nutrigenetic studies. So there's a ton of power here. It is going to become one of the premier tools in the future. Everyone will have their genetics run and understood as they navigate every one of the health and disease states that they're trying to overcome or prevent in many ways. This is a really important technology. We are certainly invested in it. We're doing it on a daily basis. We're excited about it from a research standpoint. And every day we're learning about new compounds, new metabolites, and truly the power in understanding our genetics and how it can guide us into a state of greater health, greater prevention, because each and every one of us has a unique reflection into what we eat, what we're exposed to. If our liver enzymes many of them can contain mutations that reduce their ability to detoxify certain compounds, whether that be compounds that are exogenous like pesticides or even internal metabolites. If they don't metabolize these things well through all of the detox phases, we need to be cognizant of that, and we need to alter our protective input, whether that be through supplementation or diet or exercise, etc., and avoidance as well. But once you have that knowledge is there forever. Once you understand it, this is a journey that you can keep someone on for the rest of their lives, because genetics don't change. Epigenetics do, and that is a sub-component of this, but genetics don't, so you can really start to gear your lifestyle towards the most healthy, personalized outcome possible. So that's why we've invested in this technology. That's why we've invested in the researchers and the scientists behind it. And importantly, that's the human component, but it also is very useful when it comes to the microbiome component, because it's the same tools. So we're really bringing both the microbiome and the human genetics to bear on our research. And that's where we get excited about it. And I hope you guys can get excited about us too, because there is a lot of information to come. I'll start presenting on individual genes and their impacts from a health standpoint and a preventative standpoint in the future. So make sure you join me and watch for that science to come. And I appreciate your time. Thanks.